Yeah. Guys, today our topic is generation and conduction of nerve impulse. Generation and conduction of nerve impulse. There are different kind of cells in our body. Neuron is different type of cell, which is self-excitable, which is they can conduct the electrical signals to the from one cell to another cell. They can polarize. Mean they can exchange of ions from their membrane. Okay, if you come to the reading, come to this, neurons are excitable, self-excitable. Neurons are self-excitable because their membrane are polarized in state. We will discuss it. Their membrane is polarized in state. Do you know why the membrane is a neuron or polarized? Why the neuron cell membrane is polarized? We will going to discuss about it. Okay, neurons are self-excitable. Their membrane is polarized. Neuron cell membrane is polarized. Okay, we will going to discuss about it. Yes. Why neuron cell membrane polarized? Okay, neurons are self-excitable, guys. It is self-excitable. Mean if any conduction of nerve impulses going to take place in the neuron cell, this is the neuron cell, this is cell body, axon, telodendrites, this is neuron. The axon part, if we take the cell membrane, the inside the cell there is a axoplasm, like cytoplasm, it is axoplasm. Okay, what is the membrane is about this axon. This is the cell of this neuron. This is the membrane. This is axoplasm. Why it is polarized? It is the polarity of this is -70 MV. -70 M. This axoplasm membrane is this membrane side of this ions are completely minus in level. This membrane, below the membrane, it is charged about -70. How it is maintained -70? How it's maintained -70 millivolts. Okay, this is neuron cell. Okay, there is a outer atmosphere is there. This is the outer atmosphere is there. This is extracellular fluid. Okay, this is intracellular fluid. Okay, as compared with extracellular fluid, this is intracellular fluid is minus 70 MV millivolt, minus 70 millivolt. How it's maintained? Mean negative ions are more in this inside the cell, or inside the cell negative ions are more, ions are more inside the cell as compared with outside. It is minus 70 millivolts mean outer atmosphere it is 0, 0 like ions 0 as compared with this minus 70. If it is outer atmosphere it is 100 MV, 100 MV, it is plus 30 MV. Always it is maintained minus 70 MV, minus 70 MV. How it is maintained? Always as compared with outer atmosphere. Outer mean intercellular fluid, mean this out of the cellular, this is the medium which is out of the cell, there is a tissue which is intercellular fluid. Okay? That is that is plus 70 is above than this minus 70. This is minus 70 inside the cell, they maintain the negative charge as compared with outside the cell. It is about how this is the polarity, this is the polarity, we can call polarity, this is the polarity, this is the charge which is maintained under resting potential, resting potential, resting potential. Resting potential. How it is maintained the resting potential? How it is maintained the resting potential? Okay, we will going to discuss how it is maintained resting potential. How maintain resting potential? Of neuron cell, neuron membrane, exoplasm of neuron. How it is maintained? Resting potential. This is the exoplasm in the cell, exoplasm. This is the membrane. So, this is the membrane as such as channels are there. This channel we can call sodium potassium pump, sodium potassium pump, sodium potassium pump. Okay. In this condition, there if the open sodium potassium pump, how it is open? Mean guys, just look into this. If any axon, this is the cell, neuron cell, there is a place between the cell of every neuron. There is a signal pass from one neuron to another neuron because of their potential differences, because of their potential differences, potential difference. Potential difference. In this potential difference, there is a signals pass from one neuron to another neuron. This is we can call self-excitable. Excitable. So, in this condition, these cell maintain in the resting. If there any no signal pass, we can call resting potential. Resting potential mean cell is under rest. There is no depolarization, there is no repolarization going to take place. Okay. In that condition, there are certain channel sodium ion pump. Okay. As compared with outer fluid, within the cell there are potassium ions are high. Potassium ions are more as compared with out fluid. Okay. Potassium ions are more as compared with intercellular fluid. Okay. In this condition, the sodium ions are higher than within the cell. Okay. As compared with inside the cell, there is a outside, there is a sodium ions are high. Okay. What is going on in the sodium ion pump? Okay. If the sodium pump ion open, potassium is trying to get out and sodium trying to enter in, but sodium is impermeable. The sodium channels are very low in quantity. Sodium is impermeable, mostly impermeable. Sodium is mostly impermeable. In this pump, what's going on? In this pump, if the channel open, sodium is going to out, 3 sodium ion going to out, 2 potassium ions are going to in. Okay. This is sodium potassium pump. It is active transport. 1 ATP utilized. If the sodium potassium pump open, outside sodium trying to get inside potassium trying to get outside. Yeah, I'll come to again. Sorry for the delay. Okay. This is sodium potassium pump, guys. Sodium potassium pump. What going on in this pump? There are more potassium ions are there here, because potassium ions are enter in 2 potassium ions. Sorry. If the more potassium are here, potassium is permeable. In the membrane. Sodium is impermeable. If the sodium going to out, potassium trying to in this condition is potassium is high, sodium is less in the resting potential. So, if potassium ions 3 exit out, sodium 3 exit out, potassium 2 enter in. So, negative ions are exit out more. Okay. So, the like positive ions are exit out more, does it mean the low amount of potassium, the positive charge is enter in. High positive mean 3 sodium ions mean this is the positive ion, right. So, 3 positive exit out, 2 positive enter in. In this condition minus charge will build up, minus charge will build up in the sodium potassium pump. In the sodium potassium pump minus charge will build up because of 3 sodium ions, pledge charge, 3 pledge charge going to out, 2 pledge charge coming to the cell. Okay? Yeah, this is one reason. We will find another reason why resting potential is 70 MV minus 70 MV, why resting potential maintain minus 70 MV. Okay. First reason is sodium potassium pump. Reason 1, reason 1, sodium potassium pump Okay. Why it is maintained mean, first reason is sodium potassium pump. In this condition pledge charges, more pledge charges going out, okay. In this condition sodium is 3 sodium exit out, 2 potassium enter in, 2 potassium enter in. Sodium potassium pump is one reason. Second reason, there are negative ions or negative proteins are there within the cell. Negative ions and negative proteins are there inside the cell. Negative ions and negative proteins are there inside the cell. Second reason, negative ions and negative proteins are inside the cell are impermeable. This is the second reason. First reason is sodium potassium pump. Second reason is negative ions and negative proteins are there inside the cell. They are impermeable. Mean negative charge are going to stable within the cell. Okay. Third point, important. Okay. Potassium are more inside the cell, inside the cell mean in the exoplasm than outside the cell. Mean interstitial fluid. So, potassium ions are more inside the cell as compared with outside the cell. They are permeable, guys. they are permeable, they are ready to leaving the cell, they are ready to leave the cell. Sodium ions are impermeable. They do not allow inside the cell. They do not allow inside the cell in the resting potential. In the resting potential. These are the three regions, guys. Three reasons are what? First reason is sodium potassium pump. Why the cell maintaining resting potential, that is minus 70 MV. First is sodium potassium pump. Second one is negative ions and negative particles like proteins are inside the cell, they are impermeable. They do not would not like to allow outside. Okay. Third point is potassiums are more inside the cell, they are permeable, that leads to the potassium out mainly. So, if the plus particle out, there is a minus charge build up inside, and there are impermeable of sodium ion coming to the cell. This is why there is a negative charge or build up within the cell. Okay. Yes, we are going to read about it. Yeah, just look into this. Neurons are self-excitable because their membranes are in polarized state. Polarized state means they can, the charge in the polarity, they can inter-exchange of the charges, the cell membrane. Okay? Yeah. Do you know why membrane? Neuron is polarized. We are going to discuss the different types of ions. Different types of ions mean potassium, sodium, chlorides. Okay? These minus and plus particles. Okay? Different types of ions, channels are there. Okay? Especially sodium potassium pump. Sodium potassium pump is the reason. Okay? The neural membrane, okay, different ions are there inside the neural membrane. These ion channels are selectively permeable different ions. Mean selectively permeable mean potassium is more permeable. Potassium is more permeable. Inside potassium, there are more potassiums are there inside, they are trying to get out. Mean potassium is more permeable. Sodium is impermeable. Sodium is outside the cell, they do not allow inside. Okay? This is what. Okay? When neuron is not conducting any impulse, mean if the neuron, they do not follow the signals, the next neuron, they do not give the signals, there is a synapse there. If the signals do not pass into the next neuron, that is conducting phase. If they do not conduct any signals from one neuron to next neuron, it is called resting potential. It is called resting in nature. The axonal membrane mean there is a membrane in the axon. Okay? This is axon, guys, neuron. This is, if we cut this is the axon membrane. Axon membrane is like, the axon membrane is comparatively more permeable to the potassium ions. More permeable to the potassium ions. Okay? Nearly impermeable to the sodium ion. Nearly impermeable to the sodium ion. Okay? Similarly, the membrane is impermeable to negatively charged proteins and present in the exoplasm. Yeah, charged particles like proteins are, they do not allow out. Okay? That is why the negative charge build up within the cell. Yeah, next slide. Yes. Consequently, the axoplasm inside the axon is contain high concentration of potassium. This is important, guys. In the resting potential, resting potential inside the cell there are high number of potassium. Inside the cell there are high number of potassium in the resting condition. Okay? The negatively charged proteins and low concentration ions are Na are very low. Okay? In the contrast, the fluid outside the axon contain low concentration of potassium. Outside low concentration of potassium mean interstitial fluid, outside low concentration as compared with inside. Okay? Yes, high concentration of Na and thus the concentration gradient is going to take place. Guys, what is this concentration gradient mean? This is the cell, potassium is more as compared with inside, that is a sodium is out is more, potassium is permeability is there, potassium they are trying to get out, sodium is impermeable, they do not allow here inside the cell. That is why if the pledge charge is going to out, there is a minus is developed inside the cell. As compared with outside, outside is the pledge charge if the potassium is exit out and sodium is also pledge charge. If the potassium is exit out, negative charge is there. As compared with outside, there is a more negative charge here inside the cell, that is minus 70 MV. That is what the concentration gradient is there between the outer atmosphere and within the cell. This ionic gradient across the resting membrane, this is the resting membrane, mean inside the cell membrane, across the inside cell membrane are maintained by active transport ions by sodium potassium pump, okay, which are transport the 3 sodium out in the cell and 2 potassium coming to here, which transport 3 sodium outwards and 2 potassium into the cell. Mean this is the cell, guys. Okay, sodium potassium pump, 3 sodium get into out, 2 potassium enter into in. So 3 pledge charges going to out, 2 pledge charges enter into cell, mean there is a 1 pledge charge is difference is going to take place as compared with outside and inside, that is minus 70 MV, guys. That is why it is maintained minus 70 MV. As a result, outer surface of the axonomal membrane is possess a positive charges, while inner surface is become negatively charged, and therefore the polarized, it is polarized. Okay. The electrical potential difference across the resting potential plasma membrane is called, that is resting potential. So this is minus 70 MV, we can call minus 70 MV, we can call resting potential. Resting potential. Resting potential. Okay. Yes. So next slide. Yeah. Generation and conduction of nerve impulses. Generation and conduction of nerve impulses. Okay. Reason, first it is polarized state of minus 70 MV, which is we can call resting potential. Okay. If it is, this is the cell guys, neuron cell. This is neuron cell. This is the cell membrane. Okay. The membranous layer maintain this part, only membranous layer maintain minus 70 MV. Only this part, membrane, the membranous layer. Okay. If we cut this, okay, it's showing like this, minus 70 MV. That is resting potential. If there is no signals pass, if there any no signal pass entire the cell, the rest mode is minus 70 MV. Okay. Reason, first we discussed potentiogenic pump, mean sodium potassium pump, potentiogenic, and negative ions or proteins are unable to leave the cell. Okay. Potassium is most abundant intracellular fluid and membrane at rest is more permeable to the potassium. This is most important statement guys for exam point of view. This is most important statement. Okay. Yeah. There are certain channels for the self excitable, mean in the nerve cell, neuron cells, they can excitable, mean they can pass the signals from one neuron to next neuron. There are certain channels. Okay. We will going to discuss about it. Yeah, guys, if any signals from one neuron to another neuron, if any like motor neurons or sensory neurons, they can pass the signals from sensory neurons. They can pass the signals from, like if any visible, any audible, okay, they can pass the signal into the brain from the spinal cord. Okay, there are certain neurons, they can carry information towards brain, and brain can analyze what happened. They can send the signals again to the motor neuron. Okay. How the signals are going to like transfer from one cell to another cell? There are millions of neurons, they can carry information. Mean it is neuron guys, one nerve cell, neuron. The signals which is generate here, okay, there is the gap between two cells, there is gap between two cells. So, the signals, if they can end this gap and they can jump the signals from here to here, they can jump signals here to here, they can carry information from here to here, mean conduction of information throughout the neurons. How it is going to take place? There are certain channels are useful for this information. Okay? That is nerve conduction and impulses, how they can, the channels how they can use for this conduction. First channel is leaky channel. Leaky channel. Leaky channel is they can leak the potassium, sodium inside the cell, outside the cell. Next channel is voltage gated channel. Voltage gated channel. If any electrical imbalance, mean negative charge or positive charge, if any changes in the resting potential, if any changes in resting potential, so the channel open, the mechanically gated channel, if any mechanical pressure is going to create in the cell, that leads to the mechanical channel open. Okay? Yeah. Ligand gated channel, if any chemical going to create a imbalance, mean any chemical activate the cell membrane, the channel is open, that is ligand gated channel. We will going to discuss it. Okay? Yes. Yes, this is the neuron cell. This is neuron cell. If this is the membranous layer, neural membrane, the signals always pass through the membrane. Signals always pass through the membrane, like this is the membrane. Around the membrane, the signals always pass from one neuron to next neuron. This is the next neuron, guys. The signals pass always in the cell membranous area only. So, like leakage gated channel, if the mechanical gated channel open, first mechanical gated channel, okay, how it is open? Any mosquito or any action is going to take place in our hand, just we can touch it, okay. There is a mechanically pressure on this cell membrane, okay. That is leads to the open a channel here, okay. So, if the mechanical gated channel open, there is a sodium ion trying to enter in. Sodium ion are trying to enter in. So, as usually the resting potential of the cell is minus 70 MV, right? Minus 70 MV is resting potential. Sodium is plus. If the sodium enter, the minus 70 become minus 60, right? So, voltage is different, voltage is going to change. In that condition, voltage gated channels open. Voltage gated channel open. Are open. Voltage gated channels are open. Okay. If the voltage is changed, there is a like leakage channels are there in the throughout the cell membrane, that is sodium potassium leakage. The sodium is trying to enter in, potassium is trying to exit out. This is leakage gated channel. Leakage gated channel. Leakage gated channel. So, there is a imbalance is there between the potential minus 70 into minus 60, that is the condition we can call action potential. They can accelerate potentiogenic changes, they can accelerate potentiogenic pump. Potentiogenic pump mean leakage gated channel open. In this condition, cell going to be depolarized, depolarize mean the cell signals throughout the membrane they can exit out into the end part of the neuron cell. Here there are certain channels are there, ligand gated channel, ligand gated channel, ligand gated channel. Ligand gated channel, they open any chemical release. If any chemical release any chemical release, there is a ligand gated channel open. How the chemical release mean? This is the neuron cell is end, again there is another neuron cell started. The signal, there is a between the neuron there is a place we can call synapse. Okay, there is a, like one telodendrites of the cell, they can release a chemical called acetylcholine, release a chemical acetylcholine. In that condition, there is a ligand gated channel open. Ligand gated channel are relevant to the calcium ions. In that condition, there is a, like, the membranous electrical charge is going to change. That's why the signals are jump into the next part of the axon. Next axon. Okay, this is about the channels. We will going to discuss the nerve conduction and impulses tomorrow. Thank you.